Hello, everyone. Good morning. Welcome to EMSL. So my name is Vimal, and I'm a biologist at EMSL. So today I'll be talking about how I'm using spatial omics technology at EMSL to help understand how plants respond to abiotic stress. For example, Poplar, and also under combination of environmental stress condition. So our demand for food, fiber, and fuel from plants will increase in the future, and plants are constantly challenged with environmental stress conditions, such as drought, salt, and heat stresses. And as you can see, these are single stresses, and in field conditions, these stresses doesn't occur in single form, it always occur in combination form. So when the stress combinations happen, as you can see, it adds an additive negative effect on plant growth and development. In combination stress, the plants are severely dwarfed compared to single stress condition that shows the severity of the stress. Not only the model plant, when you go to the tree model, which is Poplar, it is mainly grown for its biomass. The biomass is severely reduced under abiotic stress condition. So I want to take your attention to this graph, which kind of shows the physiological measurements done on leaf tissue. For example, they tried to measure 4 different parameters with the leaf tissue exposed to single stresses and combination stresses. As you can see, when the heat stress happens, the leaf temperature increases, the plant has to cool its leaf down, so it increases its stomatal conductance to cool its leaf. That also, it doesn't have any effect on photosynthesis, but the stomatal conductance increases to cool the leaf down. But when the plant is exposed to a complete different stress, such as drought stress, which is water limitation, the plant cannot afford to lose its water. So the stomatal conductance goes down to conserve water, but it also reduces the carbon fixation. As you know, stomatal opening is correlated with carbon fixation. So the plant has to come up with different ways to deal with drought compared to heat. But when these two stresses combine, the plant still affords to conserve water by closing its stomata so that the conductance decreases, but the respiration increases. So this kind of shows when a combination stress happens, the plant adapts a different strategy compared to single stress condition. And the omics data performed on this leaf tissue clearly shows there are unique sets of metabolites and transcripts that are upregulated in combination stress that are not found in single stress condition. So even at whole tissue level, when combination stress happens, the plant tries to adapt and comes up with different sets of metabolites and transcripts to deal with it. So when we take a granular view, for example, the recent RNA-seq experiments conducted on leaf and root shows how complex these tissues are, and these tissues are made up of several differentiated cell types with unique function. And these are all the transcripts that comprise this cell type. These are the transcripts that comprise this cell type, which shows that how complex or how unique these cell types that make up this tissue are. So this is in normal condition, when you take a normal plant and see what are all the cell types and what are all the molecular profile that make up those cell types, what happens under environmental stress condition. So when we started doing spatial proteomics, we looked at 2 main cell types of leaf, that is palisade and vascular. As you know, palisade does photosynthesis, vascular is more like a supportive and conductive cell type. We wanted to ask this question: when these cell types are exposed to stress conditions such as drought, what happens? So some of the whole tissue work performed on drought showed a set of proteins that go up and down during drought stress condition, but the orthologs of those proteins are clearly clustered in 2 cell types. For example, the upregulated proteins are clustered into palisade specific and vascular specific. Same for downregulated proteins. This clearly shows the cell types are not only unique with its differentiated function under normal condition, but also respond to the stress in a unique way. So this was the whole idea behind the CIPRO Poplar project, which is a DOE BER funded project, where we looked at spatially how plants respond to different environmental stress conditions. And then we used several omics technology at EMSL, and then we generated transcript, protein, and metabolite data at spatial scale. And we not only generated the data, we also picked candidates and developed synthetic promoters to drive these candidates in a much spatiotemporal manner. So all the stress treatments were done in UC Davis in our collaborators lab. And the 40-day-old hybrid poplar cutting were exposed to different stress conditions like drought stress, salinity, and heat stress, and their combination. So we collected samples at the start of the stress and a prolonged exposure to stress, and then we collected the second sample, which is a late stress, and then we allowed the plants to recover, and then we collected the third sample, which is a recovery. All the samples were collected and shipped to EMSL, where we performed multiomics on those samples. So initially, to do spatial proteomics, this was mainly possible because of the nanopods platform established at EMSL by Yingzhu and Ryan Kelly. So basically, the samples shipped to EMSL, we cryosection them, we laser capture to isolate the cells of interest, and then we put those cells in this nano volume droplet in this nanowell chip, where we collect about 25 samples in one chip. The entire proteolytic sample processing is done in that nano volume droplet. So basically, by putting that nanowell chip in a humidity chamber to avoid volume loss. So the entire proteolytic steps are done in this volume and then injected into the mass spectrometer to identify the proteins. And then we identified proteins from two cell types of leaf, palisade and vascular, as we talked about, and then two cell types of root, cortex and vascular. As you can see, there are unique sets of proteins that are comprised by this cell type, which gave rise to these unique pathways. For example, palisade is known to do photosynthesis, and that's what we saw it to be enriched pathway in palisade. In vascular, there were different pathways such as carbohydrate biosynthesis, TCA cycle, secondary metabolite biosynthesis. When we go to the root cortex, we saw some more unique pathways for the root cortex, such as xylem degradation, cinnamate biosynthesis, etc. But the root vascular shared some of the pathways with leaf vascular. As you know, that vascular is a continuous strand of cells that connects the above ground and below ground. So we saw some level of overlap between the pathways when it comes to vascular cell type. And we published this work in a method paper some time back. So after performing or establishing the cell isolation and cell type specific proteomics from Poplar, leaf, and root cell types, we went ahead and performed stress treated samples. So we had drought, salt, heat, and combination stress treated samples. Altogether, irrespective of stress condition and cell type, we found about 6,800 proteins from Poplar leaf cell types. These proteins were identified based on multiple peptides per protein. Do we have enough replicates, like a presence of a protein in enough biological replicates? And did the proteins cross limit of detection? So stringent parameters were used to identify these proteins. And as you can see, among the 6,800 proteins, we saw some proteins to be palisade specific and some are vascular specific, and there was an overlap. So what happens to differentially altered proteins, proteins that change in stress versus control? What we saw is a same pattern, a unique set of protein that comprise palisade that respond to any stress condition, and then the same for vascular. And the same we see for combination stress condition. There were like about 100 proteins that are unique to palisade and vascular that respond to combination stress condition compared to the control cell types. So this is between cell types. So how the cell types share and have unique sets of protein when it comes to combination stress. What about between stress conditions? Does combination stress share more with any of the single stress or not? To do that, we made this plot which clearly shows the combination stress and heat stress overlap with more number of proteins, or in other words, heat stress has more say when it comes to a combination stress than drought or salt. So, but apart from the shared proteins, we have this unique sets of proteins that are not only cell type unique, but also stress unique. In other words, the plant leaf, Poplar leaf cell types, they respond to a specific stress with a different set of proteins and associated pathways. So moving forward, these the two slides that I showed is based on fold change based approach. We also wanted to take another approach to see proteins that don't change by fold change, but change in a subtle manner, but still are important to stress response. To do that, we worked with Ryan to generate this co-abundance network. So basically, Ryan, what he did was he took the proteins that are present in drought, salt, heat, and combination, all four conditions, and then he made a co-abundance network out of it. And then when he removed a specific data, for example, when you remove a combination stress specific data, we found some proteins of high centrality shift to low centrality. And when he brought back the data, they regain their centrality. In other words, sets of protein that are more correlated with combination stress specific proteins than with others. In this way, this is called data exclusion approach. In this way, we found subsets of protein that likely respond to a specific stress, such as combination stress. So taking these two approaches, fold change based approach and network based approach, we looked into the biological pathways that are altered when it comes to single and combination stress. There were several unique pathways. As you saw in the previous slides, there were unique sets of proteins when it comes to each stress, which comprised unique pathways. For example, when it comes to combination stress, palisades is upregulating xylem degradation. What it is doing there is something to be figured out. And when it comes to drought stress in vascular, it is upregulating a protein in linoleate metabolism. As you can see, these proteins are not only fold change-wise upregulated, are highly central to the network as well. So these could be some critical proteins regulating the combination stress response. So apart from the unique pathways, we also have pathways that are common. As you can see from the name of the pathways, these are central metabolism pathways like amino acid biosynthesis, gluconeogenesis, glycolysis, etc. So these pathways are kind of shared. So this is more like a shared response to when it comes to abiotic stress. These are more like unique response to abiotic stress. So then we ask this question. So under combination stress, what are all the most significantly altered proteins? When we looked at that, these are the most upregulated protein when it comes to combination stress, but not in single stress, in two cell types, palisade and vascular. One of these proteins is glyceraldehyde 3-phosphate dehydrogenase, or GAPDH, or GAPC1. It's a cytosolic protein, and it is a central carbon metabolism pathway protein. So this protein was only upregulated or highly abundant at protein level in combination stress only in palisade, but not in vascular. And this is happening only in combination stress and not in single stress. So the poplar leaf, when it is exposed to combination stress, is upregulating this GAPDH exclusively in palisade at much higher fold change. Maybe it is important to regulate a pathway. That's what this signifies. So moving forward, we not only have the proteomic data, we also have transcriptomic data. Then we ask this question, what are all the proteins and transcripts that are highly associated with GAPDH? For us to, we wanted to get a lead from GAPDH. So we did this protein transcript association network and looked at the set of proteins and transcripts that are highly associated with GAPDH. These are all the proteins and transcripts denoted by different signs. What we saw was the other candidates in the central carbon metabolism were highly associated with GAPDH. In other words, if GAPDH changes, it's going to change the behavior or abundance of these enzymes as well. So when we put all this into a pathway map, so this is the central carbon metabolism pathway where you can see glycolysis here, TCA cycle, sugar alcohol production here. And this is the enzyme we are talking about, which is GAPDH, which is upregulated in transcript level and protein level. At protein level, we have a much higher upregulation. What about the other enzymes of the pathway? And as you saw a second earlier, so all the other candidates were also upregulated at transcript level and protein level. This signifies that the central carbon metabolism pathway is critical for palisade cells when it comes to combination stress. So this is transcript and protein. What about the metabolites? In order to study the metabolites in spatial manner, we work with Dushan and Chris, where we use the same cryosection for MALDI-MSI imaging. Whereas in the earlier spatial transcriptomics and proteomics, we take the cryosection and isolate the cell of interest for doing the omics. Here, we don't have to isolate cells. We use the actual cryosection to do MALDI-MSI imaging, which spatially locates metabolites. For example, here there are two metabolites, how they are changing in control condition and stress condition. For example, one of the metabolite is increased in palisade cell area, but not in vascular area. Whereas the other metabolite is changing in vascular area, but not in palisade area. So this kind of shows the importance of this technique to study spatial abundance of different metabolites when it comes to abiotic stress condition. So going back to our pathway, we tried to map different metabolite abundance. So for example, here is the metabolites that are linked with GAPDH. For example, as you can see, these two charts here, one of the metabolite is upregulated in palisade, whereas the other one is reduced in palisade. This one being upregulated, it kind of shows the pathway is going in the reverse direction, not towards TC cycle, but the other way. If this is going in the other way, because this is a reversible reaction, if the pathway is going the other way, it could be gluconeogenesis to make sugar and sugar alcohol to serve as a function of osmolyte during stress. Then we measured the level of sugars, which is hexoses, which is increased in palisade cells, and one of the sugar alcohol was also increased. So this clearly signifies, along with the transcript and protein data, that palisade cells, when it comes to combination stress, could be activating gluconeogenesis to make sugar alcohol. In order to confirm this metabolite data, we also did a complementary GCMS-based metabolomics, where we collected the palisade cells and sent for GCMS metabolomics, which identified most of the sugar alcohols to be elevated in abundance in palisade cells, and some of the TCA cycle compounds as well. So this shows this pathway is activated to increase sugar alcohol production during combination stress. Going forward, we are trying to prove this using two different approaches. One is to infiltrate Poplar leaf with stable isotopic fatty acids. So this is the image of an UV fluorescent dye injected into Poplar leaf to show how efficient is our infiltration process. So right now we are infiltrating with a stable isotopic fatty acid, and then we are exposing the poplars to combination stress to see if this signature from the fatty acid is transported to the sugar-sugar alcohol. In that case, we can confirm that this pathway of gluconeogenesis is activated during combination stress in palisade cells. Along with that, we are also trying to overexpress our candidate GAPDH using a synthetic promoter. So this synthetic promoter is designed by our collaborators, basically by using the cell type transcriptomics data that we generated. So they mined for the motifs that are highly enriched in the cell type transcriptomics data to generate a promoter that can be strongly activated under stress than the native promoter. So right now we are trying to use this strong promoter to drive our candidate in Poplar. And the aim is to generate the transgenic Poplar and expose them to combination stress and see at palisade level what are all the physiological parameters or molecular responses improved. So these are the two approaches right now we are taking to understand how Poplar respond to combination stress. So this project kind of shows how we used spatial multiomics facilities at EMSL to try to understand how plants respond to combination stress. I want to give a quick snapshot of the recent development when it comes to proteomics. So right now we are moving forward from cell type based proteomics, which is a few hundred of cells we collect using LCM. Right now we are moving towards single cell based proteomics. So basically we are trying to isolate the protoplast from the leaf and then sort them and then put them on the same nanowell chip and do mass spec to do the protein abundance from this protoplast. So what we have right now is from a single protoplast, we have about 2,000 proteins detected. And then we also conducted an experiment where we had a control leaf and a drought leaf to see how at single cell level, the proteins change when it comes to drought stress. What we see in one of the stress conditions, which is severe stress, there are groups of protein that are different, or the cells are behaving different compared to the control cells, showing that these cells are activating some of the enriched pathways under drought. So these are all the pathways that are enriched at a single cell level. So we generated single cell proteomics data under drought, but we wanted to compare with what is out there. So when it comes to single cell, the most commonly used approach is single cell transcriptomics. But the advantage is they do single cell transcriptomics and then they use marker genes to identify clusters of cells from leaf. So we wanted to see what are our proteins matching to. Interestingly, our proteins match the highest with the clusters that's called mesophyll cells, which is kind of telling that what we isolated is mesophyll cells for proteomics. And we not only compared with single cell transcriptomics, we also compared with whole leaf proteomics. So in this case, we have single cell proteomics. This is whole leaf proteomic data. We have an overlap with whole leaf proteomics to some extent, but also we have unique proteins. So as you know that when we do a whole leaf based analysis, the response that we get is an average response of multiple cell types. So maybe by doing a single cell proteomics, we are seeing a unique response coming from one cell type that may give us some unique candidates. So here is where we are. The method is established and it's available for EMSL users to study underrepresented cell types and how they behave to a specific stress condition in the future. So with this, I'll thank my postdoc advisor, Amir Akami, and all the colleagues who really worked hard as part of this project. I thank the collaborators for healthy discussion to help develop a biological story out of the multiomics data. And then I thank the funders, DOE, BER, and I thank EMSL for all the facilities. I thank you all for listening.
Really great talk, Vimal. Thank you very much for giving that. I think we have time for 2 questions before the coffee break today.
A very, again, Peter, Berkeley Lab, a very practical question. How long does this actually take to do these experiments? Let's say that you get your cells, like what kind of timeline are we looking for you to actually have data? How does this work?
So, so for spatial proteomics, we cryosection them, LCM capture the cells of interest, and then we give it to the proteomics team. And depends on their queue, it can be run either faster, which can be at the early fiscal year, things can move really faster. If you get your samples in early, things can go very faster. That's what I can say. But the turnaround time can be, depending on the availability of the instruments, it can be in a matter of days. In days, yeah, matter of days. If the instrument is available, all the cryosectioning, LCM, and the proteomics can be done in a matter of days. or matter of couple of weeks or few weeks.
That is days for a single leaf with multiple punctures, or what if I have 100 plants just being wild here?
Okay, for the 100 plants, looking at one cell type, the bottleneck would be the LCM. So the laser capture process is a little time taking to cut out enough number of cells. For example, here for Palisades, we had about 500 to 1,000 cells. It takes about 3 to 4 hours to cut one biological replicate, one leaf, one cross section. So I think that is the bottleneck here.